Good evening, everybody, and welcome to tonight's Public Insights lecture. My name is Sally Shortall. I co-chair the Public Insights Committee and I work in Newcastle University. And I'm really pleased to introduce our speaker for tonight, Professor Deborah Tweddle, whose title of her talk is Neuroblastoma, a Master of Disguise and a Challenge to Cure. I will be back after Deb's lecture to host a live Q&A with Deb. If you would like to raise a question, you can do it through the chat box function on YouTube, or you can tweet us. So if you want to tweet us, it's @InsightsNCL. If you want to tweet about the lecture while it's happening, please use the hashtag InsightsNCL. So here to more completely introduce Deb is Professor Andrew Blameyer. So enjoy the lecture and I'll see you afterwards for a live Q&A.
Good afternoon. My name is Professor Andrew Blameyer and I am the Dean of the Translational and Clinical Research Institute in the Faculty of Medical Sciences here at Newcastle University. It's my pleasure to welcome you to this virtual Insights lecture, which is part of a celebration of 40 years of life-saving cancer research here at Newcastle University. Cancer research in Newcastle started back in 1982 with a very small seed of only 7 principal investigators and has since grown into a large and vibrant research community, which today numbers more than 600 individuals and spans the university and the NHS trusts in our city and our region. In 2019, we acknowledged the growth and the importance of cancer research within the university by incorporating a new University Centre of Research Excellence in Cancer, which now leads all of our research in that area. Our research is world-leading and is recognised as making major and important contributions to both the fundamental understanding of cancer and to improving diagnoses, to developing new treatments, and ultimately to improving the lives and the survival of people living with cancer, both in the region, in our country, and indeed worldwide. Our speaker today is Professor Deborah Tweddle. She's an Honorary Consultant in Paediatric and Adolescent Oncology at the Great North Children's Hospital, and she's also a Professor of Paediatric Oncology within the university. As one of our clinical academics, she divides her time between leading her academic research team and undertaking a wide variety of clinical duties, including looking after patients with neuroblastoma, working as the scientific director of the Children's Cancer and Leukaemia Group Tissue Bank, and also acting as a co-director for the National Neuroblastoma Genetics Reference Centre service, together with her colleagues in the cytogenetics service within the NHS. Professor Tweddle's research interest is in childhood neuroblastoma, and it's an area where she's been specialising in for over 20 years. So she's going to take that as part of her topic for today, and she'll be speaking on neuroblastoma, a master of disguise and a challenge to cure. So I'll now hand you over to Deb, and I hope that you enjoy today's lecture. Thank you.
It's a pleasure to talk to you all today about my chosen subject, which is the clinical conundrum that is neuroblastoma. As already mentioned, I'm both a clinician and a scientist, and I look after children with neuroblastoma at the Great North Children's Hospital, and I have an active research lab within the Wolston Childhood Cancer Research Centre. So before I continue, I just want to mention that this year marks 40 years of cancer research at Newcastle University. And it's also 20 years since I've been studying neuroblastoma research here at the university, and 20 years, in fact, that Cancer Research UK has been in existence. So today, what I'm going to talk to you about initially is the clinical presentation of neuroblastoma and how we stratify it according to risk groups, which then determine treatment. Intertwined within this is the importance of the genetics of neuroblastoma and how they help us to refine risk groups, and perhaps more importantly, how they've led to new advances in treatments over the last 20 years. And I'll provide some examples of that. And lastly, I want to finish by telling you what I think may be the future ways in which we will proceed with neuroblastoma research and treatment over the next 20 years. Neuroblastoma is the commonest solid tumour of childhood outside the central nervous system. In fact, it's the commonest cancer of all children under the age of 1 year, so that's commoner than leukemia. It's a tumour that arises from the sympathetic nervous system, which runs either side of the spinal cord from the neck down to the groin. But the commonest site for a neuroblastoma to develop is here within the adrenal medulla. And approximately half of all tumours present with an abdominal mass, which originates from the adrenal gland. But as you can see, the sympathetic nervous system, because it's either side of the spinal cord, can run from the neck to the groin. So patients can present with tumours in their neck, right through into their groin area. And I'll explain the presentation of some of these later. It's a tumour of young children, with nearly all cases, 90% being present below the age of 10 years of age. And the median age of presentation is around 18 months. And because of that, it's one of the embryonal malignancies of childhood. And under the microscope, what you will see is these small round blue cells, which on their own can't tell you which of the embryonal tumours it is, but by other special stains you can. And in particular for neuroblastoma, you often see this angel hair or neuropil, which reflects the neurological origin of the tumour. I'm pleased to say that over the last 30 or 40 years, we've made huge advances in the treatment of neuroblastoma. And as you can see from this survival curve, where it shows a proportion of patients surviving versus the time at which they were diagnosed, there has been significant improvement from less than 40% survival of all patients with neuroblastoma in the late 1970s up to the end of 1980s, through to 2010 onwards, where nearly 65% of children are now surviving neuroblastoma. Although this is a significant improvement, it isn't nearly as good as some other childhood malignancies, and particularly leukemia and lymphomas enjoy in excess of 90% 5-year survival. And this is what we are really striving towards with neuroblastoma. But in fact, there are some patients who really do very well. And what we do is to refine neuroblastoma according to risk groups. And by this, what I mean is patients who are likely to have a good outcome versus those who need more intensive treatment to achieve a better outcome. So this Kaplan-Meier survival curve shows the neuroblastoma cohorts separated according to risk group, with patients in the low and intermediate risk group having greater than 90% survival, whereas patients who are in the high risk group, their survival is currently still only around 50%. Just to expand a little bit more on these risk groups, how do we define low risk? Well, these are patients with localized tumours. That means tumours that haven't spread, or patients who are under the age of 18 months, so under the median age of diagnosis of neuroblastoma, or have a special type of metastatic spread of disease, which I'll come on to later. Similarly, the intermediate risk or medium risk group, these patients also have a good outcome on the main, and these patients can be metastatic if they're under the age of 1 year, or they can be patients who have localized disease, so disease that hasn't spread, and those patients are over the age of 18 months. Together, both these groups account for half of all neuroblastomas. The main group, however, is the high-risk group. So these patients have either metastatic disease that's spread to distant sites such as bone and bone marrow over the age of 12 months, or they have disease that's MYC-N amplified, and I'll come on to what MYC-N is shortly. And as a result of the intensive treatment that they have, we can cure, as I mentioned, around half of these children, but this is what our research efforts are most directed against to try and improve the outcome for this group of patients. So I'll now turn to the genetics of neuroblastoma. So by genetics, we're referring to, in the main, the genetics of the actual cancer rather than genetics of every body cell, which is called germline genetics. So if you imagine in all our cells, we have a cell nucleus, which is where the chromosomes are stored. And there are 23 pairs of chromosomes together with an X and Y chromosome, depending if you're male for X and Y or female for 2 X's. And the chromosomes are all made up of a long arm and a short arm. The short arm P stands for petite, and the long arm, which is what the genes are wrapped around in DNA, which makes up structures which we can then use to determine the genetic code, which is made up of bases A, G, T, and C. And these genes are what are really at the nuts and bolts of what cancer abnormalities are, because cancer is a genetic disease. So if we turn and focus on particularly the MYC gene, which, as I've spoken about, causes high-risk neuroblastoma, what we do with neuroblastoma tumours is to test for this using a particular technique called fluorescent in situ hybridization. And MYC being an oncogene, or any gene in the cell, should usually be present in 2 copies for each of the different chromosomes. And so 2 copies would be present on chromosome 2. But in neuroblastoma, where the MYC gene is present in many copies, you can see here that in this slide where you make an imprint from a tumour on a slide and you bind a fluorescent probe to it, you get many, many more than 2 copies of the MYC oncogene. And as a result of this, this makes the tumour fall into a high risk group and need more aggressive treatment. As I mentioned, the normal cell should just have 2 copies of MYC, and then which is in red here, and 2 copies of a green reference probe. And this test we can do very quickly on all neuroblastomas and get results back to the referring clinician within 2 or 3 days from around the country. And that can immediately determine whether or not the patient falls into the high-risk group. However, we've known about MYC for some 30 or 40 years now, almost 40 years actually. And so really we've been studying the genetics throughout this time to find other important genetic prognostic markers. And as a result of many efforts from many labs over the last 20 or 30 years, we've realized that many other chromosomes are involved, and not just chromosome 2 to P where MYC is. And one of the ways we can look for the chromosomal abnormalities is what's called a single nucleotide polymorphism array. And I've depicted this in this diagram here, which shows that when you extract DNA from the sample, so this isn't a tumour imprint as was done for the fish, when you extract DNA, you can add it to one of these SNP arrays, which then binds the DNA to a normal DNA sample. And where the DNA is present in extra copies on chromosomes, there's strong binding where there's a duplication of that particular chromosome or part of the chromosome. And where there's a loss of that particular chromosome or part of the chromosome, you get weaker binding. And this is probably now the single most important laboratory test for the genetics of neuroblastoma, apart from MYC, as I'll show you, leads to further risk stratification. And so as a result of doing SNP arrays, we've been able to produce these images from the SNP arrays, which can tell us whether or not there's 2 copies of a chromosome, as can be seen here on this graph where the line is at 0, or whether there's gain in a copy of a chromosome where there's 4 tracks on this lower graph, or where there's a loss of a chromosome where the number of tracks reduces. And this tells us for all the chromosomes whether there's loss, gain, or whether there's the normal 2 copies, which is called diploid. And so to give you an example of what this looks like, because it's so important in neuroblastoma, I'm going to highlight it. The first upper panels, the upper 2 panels, and each of the chromosomes is represented here by a different colour using the software for the SNP arrays. So we start from chromosome 1 up to chromosome 22, and we find that where there's changes in the entire chromosome number, that there's an increase in copies here or a reduction. So in this particular profile, there's a few genes that are the normal 2 copies, but many genes have got additional copies, many chromosomes rather, because these are whole chromosome losses or gains, and in this example, whole chromosome gains. Whereas in the example given below, what I've highlighted is rather than whole chromosome losses or numerical copy number abnormalities, we've got losses or gains of parts of chromosomes. And we've learned over the last 20 years that there are particular genes, particular segments of chromosomes that are important in neuroblastoma. And these are outlined here, particularly chromosome 1p, 3p, 4p loss, or 11q loss, or gain of 1q, 2p, or 17q. And in this figure, you can see that there's chromosome 3p loss, there's chromosome 11q loss, and chromosome 17q gain. And that tells us an awful lot more about neuroblastoma, as well as the ability to look for extra copies, so amplification of genes other than MYC-N, as I showed you before, but other particular oncogenes or cancer-causing genes, which may be important therapeutically. So to then use the genetics within the risk groups, what this slide shows is that for patients that have low and intermediate risk neuroblastoma, many of them will have the genetic profile of whole chromosomal abnormalities that I showed you before. And this is a really good genetic marker, because this tells us that the tumour has favourable biology, and as a result of these changes in whole chromosomes only, there's a good chance that if the patient falls into a category of being low risk from being quite young, so often in babies, there's a very good chance that these tumours will go away by themselves or need minimal treatment. So for low-risk neuroblastomas, and this is absolutely fascinating, that some tumours can undergo spontaneous regression if they have this good biology feature. And what happens is that in young babies, many of these tumours can be picked up just by accident or with minimal symptoms. And if we have the biology as shown here, we can just observe these tumours on MRI scans. However, there are some infants that are diagnosed with neuroblastoma where, because of its location, and as I showed you earlier, it can be either side of the spinal cord, it actually invades into the spinal canal and causes weakness of legs. And those tumours, because of the paralysis that's resulting from the location of the tumour, will need some chemotherapy in order to stimulate the process of regression. But in fact, that chemotherapy can be quite minimal and just enough to relieve the spinal cord compression and then be able to go away spontaneously. Similarly, there are cases that rather than actually regressing spontaneously, the tumour matures as a result of its good biology. And I show that here in this picture, where the tumour's undergone maturation, where these cells have become very large, called ganglion cells. And these cells then are becoming benign rather than cancerous, and will just then remain in the patient's body just as a benign structure, and usually don't cause any further symptoms. The spectrum of neuroblastoma is such that as a result of presenting anywhere from the neck to the groin, you can get a neuroblastoma presenting in the neck, which can cause this abnormality shown here, which is something called Horner's syndrome, where patients have different coloured pupils as a result of a neuroblastoma within the sympathetic chain of the neck, where during early childhood it's affected the normal coloration of the iris. So you can just see subtly here that this patient's got one green eye and one blue eye, and the blue eye is associated with a slightly smaller pupil and slight drooping of the eyelids, something we call a Horner's syndrome, which is a well-recognised feature of neuroblastoma. The intermediate risk neuroblastoma can present similarly to localized neuroblastoma, but also with features of metastatic disease in infants, which still has a very good outcome because it's the older patients with metastatic disease that have the high risk disease. So overall, the outcome for low risk disease is really very good. We stratify, as I've mentioned, the patients by age and by their tumour biology, and if they have symptoms. So those patients who are young with good biology and without symptoms can be simply observed, whereas some patients will need more treatment depending on the biology and depending on whether they have symptoms. So there's a whole raft of ways in which low risk neuroblastoma can present, from a baby with a very large liver and skin nodules, something that's called 4S or MS appearance, to being a localized symptomatic or asymptomatic mass, Horner's syndrome, which we've talked about, spinal cord compression, something called opsoclonus myoclonus syndrome, or dancing eyes, dancing feet, which is due to antibodies produced against the neuroblastoma that cross-react with normal nerve cells in the eye and in the cerebellum, so they affect balance, so the patients can be unsteady. And also we pick up neuroblastomas because it's the commonest cancer in babies as a neonatal mass, which is diagnosed even antenatally before the baby is born. And usually we can just watch these tumours because they actually have a very good chance of spontaneously disappearing without even need for biopsy. So we now contrast that with what it's like to have high-risk disease, and this is the other half of all neuroblastomas. And this group, as I mentioned, are patients over the age of 12 months. They have metastatic disease to distant sites, including, as you can see in this slide here, in the bone marrow, it's the bone marrow aspirate showing neuroblastoma, or classically bony disease, distant bony disease, and this is a special scan called an MIBG scan, meta-iodobenzalguanidine, where the isotope is taken up in areas of neuroblastoma throughout the body, and often there's quite a number of these bony metastatic sites of disease in high-risk disease. And also, as I've mentioned, the biology is such that the patients in half of cases will have amplification of the MECAN oncogene, as shown again here by this fish slide. And this often marries up with what you see down the microscope on a biopsy, where the tumour is what we call undifferentiated, where you can't see the neutrophil we saw in that first slide, but the cells are undergoing a lot of cell division, and as a result of having the MECAN amplification, the tumour is rapidly proliferating. If we contrast the favourable SNP array of whole genome chromosome gains and losses with this pattern, which shows, you can see these spikes here, which represent amplification of oncogenes such as MECAN, ALK and MDM2, and then this overall sort of jumpy appearance of losses and gains of segments of chromosomes rather than the whole chromosomes, which portend a good outcome and are a favourable biological feature. So how do we treat high-risk disease? Well, for 15 years, we treated patients in the UK and around Europe on the European high-risk neuroblastoma trial. And I won't go into a lot of detail, but this roadmap shows you essentially the main features of treatment of all cancers, not just neuroblastoma, where you begin with remission induction. So this is a combination of different chemotherapies to try and remove as much metastatic disease as possible. And that then enables a patient to proceed to the second phase of treatment, which is surgical resection of the primary tumour, which is often in the adrenal medulla, as I've mentioned earlier. And then following this, the patient can have what's called consolidation of their remission, where you aim to maintain the patient within a remission period by giving high-dose chemotherapy, sometimes called myeloablative therapy, because it wipes out the bone marrow, and the bone marrow needs to be rescued with stem cells. And this is a much higher dose of chemotherapy to try and get rid of any remaining malignant clones. It's quite aggressive treatment. And following that, the patient then has further consolidation treatment with radiotherapy to where the primary tumour was, since it's now been removed, and finishing up with a final phase of treatment called minimal residual disease phase, which involves giving treatment for around 6 months with 2 regimens of giving an agent to a mature neuroblastoma cells called 13-cis-retinoic acid, but importantly, a completely different way of giving treatment for cancer using immunotherapy against an antigen called GD2. And anti-GD2 immunotherapy has been around now within the UK and Europe for around about 15 years. It was introduced approximately halfway through the high-risk neuroblastoma trial, and it was given, initially planned to be given with what are called cytokines, so some growth factors. And so our main aim of our trial was to give this treatment at the end of the other forms of treatment, such that the immunotherapy would act by a completely different mechanism. So you can see in this cartoon diagram that here is the GD2, which stands for disyloganglioside, a sugar lipid molecule on the tumour cell, and what are given is an antibody against the GD2 in the same way as you would hear of antibodies against COVID. And these antibodies bind to the GD2 on the tumour cell and recruit the normal white blood cells, the other types of inflammatory cells, and they make the cell undergo different form of cell death than you would have from having chemotherapy, something called antibody-dependent cytotoxicity or complement-dependent cytotoxicity. And when we first studied GD2 immunotherapy in neuroblastoma, we didn't know whether we would be successful giving it on its own or whether we needed these cytokines. And the importance of these cytokines is that they can increase the numbers of white blood cells that are involved in this process, but they also have many more side effects than if you were to give anti-GD2 immunotherapy alone. So it was important to find out whether they were really necessary. And I can share with you the result of one of the arms of the high-risk neuroblastoma trial, something called the R2 randomization, which was comparing patients that received anti-GD2 alone versus those receiving anti-GD2 and subcutaneous interleukin-2 as one of the cytokines. And this paper showed very clearly that for patients who received the anti-GD2 with the cytokine, with IL-2, their outcome was not superior to those who didn't receive the cytokines, whereas they had much more toxicity. And as a result of this, we now recommend the anti-GD2 is given without any cytokines. And in fact, that's how it's been approved and is now standard of care for neuroblastoma worldwide at this phase of treatment. I'm just going to pause here to say that although I've gone through this quite rapidly, actually high-risk neuroblastoma is a very challenging condition to treat, and it can be quite a roller coaster for the family and a journey that can take them in many different directions. So I share this cartoon from one of my colleagues, Sue Carey, a Young Lives versus Cancer social worker here at the Great North Children's Hospital in Newcastle, who depicts what it's like for families to be diagnosed with high-risk neuroblastoma, where their journey begins following diagnosis with chemotherapy, and hopefully it proceeds to surgery, but sometimes there's a whole raft of other treatments needed at this point before surgery in order to get the patient into remission. However, all being well, they then proceed to high-dose therapy, and that can come with many complications from the high doses of the drugs. But again, doing a trial has shown it's very successful, and so we do continue to give this, but sometimes there can be problems and it is intensive treatment. But all being well, patients then move on to radiotherapy, and then they move around to immunotherapy. And fortunately, it's at this point of immunotherapy or just beyond then that around half of patients sadly will actually progress or relapse from their disease and then enter a journey where they will try to undergo further remission, but usually it tends to be controlling the disease at this point rather than curing it. However, those that the 50% that we can cure currently, they will then go on to lead a relatively as normal as possible post-cancer quality of life, and usually get back to school and attain a good quality of living without a huge number of late effects. Although we know, because children are surviving high-risk neuroblastoma, that we do need to be aware of these late effects of treatment and try and target our treatments in future to try and reduce some of these by giving more targeted treatments, which I'll come on to. So this brings me to some work that we did last year with a gene called anaplastic lymphoma kinase. As its name suggests, it was first discovered in anaplastic large cell lymphoma. It was also found to be important in other cancers, not just neuroblastoma, but some work around sort of 12 years ago now identified it as an important gene in neuroblastoma. So what it is it's a kinase, means a protein that phosphorylates, leading to cellular downstream pathways leading to cell growth, and ALK sits on the cell membrane where it receives binding from a molecule which would normally stimulate it in a wild type normal setting, and that would then lead to pathways for cell growth. And it's particularly involved in normal neuronal development, so normal development of the central and peripheral nervous systems. But in cancer it can be altered such that it's in a mutant form, so in a way that that we've become familiar with COVID mutations. And in this scenario, the protein is active all the time, so it's stimulating cell growth, but also it can be active from being present in lots of copies in the same way as we saw that with MYC earlier. So ALK amplification also leads to rapid growth. And so what we've learned over the last 10 or 12 years is that ALK is a very important gene in neuroblastoma. It's mutant in around 10% of cases, and it's also the commonest germline mutation, meaning that for those 1% of neuroblastomas that are hereditary, ALK is the commonest cause. Around 60% of those will have a mutation present in every cell, not just the cancer cell. We've learned from studying ALK that it's present in all the risk groups that I've told you about earlier, and that it increased frequency of relapse occurs, of mutation occurs at relapse. And we now know it's a prognostic marker in certain types of neuroblastoma, particularly high risk, as I'll come on to later. And as a result, ALK inhibitors, so small molecule inhibitors of the ALK protein, are in early phase clinical trials for neuroblastoma, and will be later coming in for high risk neuroblastoma. So we test ALK using a number of different means, particularly by sequencing the gene, which we can now do using a rapid method within Newcastle in our Neuroblastoma Molecular Diagnostic Reference Centre service. And we can also test for amplification using a SNP array, as I showed earlier. This slide shows the study that we carried out last year as part of the high-risk neuroblastoma trial within CEAPEN, and we looked at a large number of neuroblastoma cases for the ALK gene within the tumour, and found that those that particularly had amplification of the ALK gene had a very poor outcome, and those that had any kind of ALK alteration, so mutation or amplification, had a poor outcome compared with those that didn't. And these were all patients treated on the high-risk neuroblastoma trial that I showed you earlier. Perhaps what was most important was that in patients who were treating on what we would now call the gold standard treatment, so those that had immunotherapy as well on the trial and had the best of the other randomizations for high dose therapy, they also had a poorer outcome. So this line here shows patients that had an ALK alteration versus those that didn't, in those that received immunotherapy. And it was really this that made us decide that actually now was the time to introduce an ALK inhibitor to patients with neuroblastoma who had an ALK aberration at the diagnosis stage of their neuroblastoma, rather than waiting until later on. And so within the next high-risk neuroblastoma trial, all patients with an ALK aberration, either a mutation or amplification, will receive an ALK inhibitor in addition to current treatment. So I just want to finish off by two other advances that are going on within neuroblastoma. And one relates to the study of circulating tumour cells or disseminated tumour cells, because these are found very commonly in neuroblastoma because it often metastasizes to bone or to bone marrow. And what we did a couple of years ago was do a study where we were able to collect blood and bone marrow samples from patients with neuroblastoma from 5 centres in the UK and analyse them using this special bit of equipment called an imaging flow cytometer, where you could measure the numbers of such cells and also their characteristics. And by labelling the cells for GD2, which I showed you earlier is also used as a treatment strategy, we could decipher what were neuroblastoma cells versus what were normal white blood cells, which you would see also within the bloodstream. And looking at these patients, many of whom were on the high-risk neuroblastoma trial, we could see high levels of circulating tumour cells and high levels of the disseminated tumour cells in the bone marrow. But perhaps what was most interesting for us was that for patients who were diagnosed with high-risk neuroblastoma and had high levels of circulating tumour cells, so high numbers within the blood, these patients were less likely to undergo a bone marrow complete response after treatment on the high-risk trial initially, compared with those those patients who had higher levels were less likely to respond to induction therapy in the bone marrow than those patients that had lower levels of circulating tumour cells, meaning that we could potentially use this in the future to predict which patients will respond to chemotherapy in terms of their bone marrow response. And so more work will be done on that within high-risk neuroblastoma too, and also not just for the cells, but for the circulating tumour DNA from these patients to be studied as a much more non-invasive marker of ways that we can do genetic testing and also monitor disease. And then my final slide is to show that there are many advances continuing with the genetics of neuroblastoma and indeed all childhood tumours, which can now undergo whole genome sequencing as part of the NHS whole genome sequencing for childhood cancer. And you can see that in these sort of very complex genomic aberrations that are present, particularly in relapsed neuroblastoma, these whole genome sequencing data can, we hope, provide additional information to what we would have normally seen on a SNP array. And by getting this more in-depth genomic information, we hope that this will lead to eventually devising new ways of being able to identify the most high-risk cases, but also provide the opportunities for new treatments. So in summary, where do I see the next 20 years? Well, I really believe that for high-risk neuroblastoma, we now need to be stratifying treatment on the basis of the genetics more so than we're doing currently. So those ones that are MYC amplified, I think, should have particular treatments that work for rapidly progressing neuroblastoma versus those that are more slow growing, which tend to be in older children with other genetic abnormalities, where perhaps a more gentle treatment but given for longer might be more effective. I also think that in line with other malignancies, giving targeted treatments such as the ALK inhibitors, which I've referred to earlier, or immune therapies such as anti-GD2 therapy alongside conventional treatment will really help to optimize the number of patients that respond and can achieve long-term cure. I've also mentioned that from the genetics, I think we can monitor disease using circulating tumour DNA in a way that enables us to predict early relapse and maybe intervene earlier, whereas before we often had to wait for it to be visible on imaging. And then I think that eventually, in the next 2 to 5 years even, whole genome sequencing will replace some of the other testing methods that I've shown you earlier. And I would like to think, as a result of the refined genetics, we can pull back even more on the treatment of patients within the low and medium risk groups than we're doing so already, if their biology is very good. And that will lead to significant reduction of late effects within this group, but also that we're tailoring the treatment much better to those with high-risk neuroblastoma, and hopefully by giving tailored treatment, we might reduce the late effects within that group as well. So finally, I just want to end by showing this picture of Cragside, where Lord Armstrong lived, who discovered hydroelectric power, and what he observed about however high we climb in the pursuit of knowledge, we still see the heights above us. And I think the more we know, particularly about neuroblastoma, the more conscious we are of how much there is still to learn in the future. So I finally want to thank all the people that have been involved in this work, both within the laboratory, past and present members that have undertaken the research within my lab on neuroblastoma, but particularly my NHS cytogenetics colleagues who've maintained a service for detecting neuroblastoma in all paediatric tumour samples from around the country for the last 20 years, as well as the research and the nursing team, and of course the patients and families who donate their tissue for research. And these are some of our collaborators within CEPEN and the many, many funders that have supported this work over the last 20 years. So thank you very much.
Deb, thank you so much for a really interesting lecture and really nicely delivered. So thank you. And I'm sure there's going to be a lot of interesting questions to come. And I'm going to start with Jonathan, and this is quite a specific question. He wants to know, can you give us an idea of how many children are diagnosed with neuroblastoma every year? And I don't know if you'd even have figures for the Northeast, but I wonder if you could give us an idea.
Yeah, so in the UK, there's around 100 children a year diagnosed with neuroblastoma. And of those, around half will have the high risk type I was focusing on. Within the Northeast, we would have between about 5 and 7 new cases of neuroblastoma each year. And again, about half of those will be the high risk type.
Right, okay, thank you. Okay, Michelle is interested to know what made you want to work in the field of cancer research.
Oh goodness, that takes me back to medical school. So I went to medical school in Manchester, and I used to live in Withington, and I walked past the Christie Hospital on my way home from lectures, and I thought, wouldn't it be fascinating to work on cancer? And at the time that we were doing biochemistry, we were learning about oncogenes, which I referred to in the talk, and I thought oncogenes, cancer-causing genes, wasn't that amazing to learn what caused cancer and how we might be able to develop new cures? And that was really what led me to initially do research at the Patterson Institute in Manchester, and then later on by coming to Newcastle as a qualified doctor and carrying out neuroblastoma research because of the strong track record of neuroblastoma research here.
Fascinating. Mavis wants to know, how have incidences of childhood cancer and survival rates changed over the course of your career?
Yeah, well, I'm really pleased to report that they have improved enormously during the time that I've been a doctor. I mean, initially when I first came to Newcastle from the US back in 1995 to undertake neuroblastoma research, we could only cure about a quarter of children, 25% of children with high risk neuroblastoma. And now to be able to cure half is really, really significant. But of course, we've still got a long way to go. We want to get to 75% or beyond, like for some other childhood cancers. And so I'm pleased to say we're making good progress, but there's still a lot more work to be done. As far as the incidence is concerned, I don't think it's really changed. Perhaps we're slightly better at detecting cancer, and we're more likely to be accurate in our diagnoses, but I'm not sure the actual incidence, the number of cases of childhood cancer has really increased over the last 20 years or so.
Okay, the question has just come in from Gemma, and I was about to ask you, is there adequate investment in this type of research? And Gemma wants to know, what are the main obstacles impeding the further advancement of treatments for neuroblastoma patients? So I guess my question's kind of tied up on that one.
Yeah, I think a lot of it is partly because it's such a rare disease, and so we need to work collaboratively. We need to all be doing clinical trials where we can invest in the patients that we see in our country versus those that are seen elsewhere, so that we can produce the maximum benefits. I think it's difficult with childhood cancer because it is so rare to get the numbers of cases to study to make those advances. But I think in terms of investment, there are charities such as Cancer Research UK, other name charities that are putting in resources to help find new treatments for childhood cancer. But I think one of the most important things is for us all to work together collaboratively around the globe. And so there's nothing better than to be able to, email your colleagues in other continents and to get advice either about treatment or about where the next thing is going in research, so that we can really, provide that much needed extra energy to be able to find new treatments and new cures, because there's no doubt it's a chameleon of cancer. It's you might have a new treatment, but in fact neuroblastoma is so clever, it comes up with ways to be able to resist those treatments. And really we've all got to be working together to try and come up with the best combinations that can try and put patients, as many patients as possible, into remission first time around.
Okay, great, thank you. Okay, and the final question comes from James, and it's a more personal sort of a question. He wants to know, can it be emotionally difficult seeing such young patients and their families being so at their weakest and most vulnerable?
Yes, is the answer. Yes, it can be very challenging. What we find is that there's such a lot of treatment that these children go through, and the disappointment to see the disease come back when you've really tried all these different treatments is really really disheartening. And one of the things I think that's helped me to continue working in this field is the fact that, you know, I can see the patients on the ward and you know be very upset by the fact that their disease can't be cured. But equally, I'm able to come back to the university and then say, well, what is it about that child, that particular child's cancer? Why did it occur? Why did it recur? Is there something about the genetics? Is there something that we can then, you know, look into in the future to try and prevent that happening in other children? And so to be able to have this role where I spend time doing clinical work and research, it is very rewarding to feel that hopefully by doing the research, you can make a difference to the next generation of children that are diagnosed and treated, and you can see where the most important research questions are to ask.
Thanks, Deb. And having said that was the last question, so many more interesting ones have come in. I'm going to take one more, and it's from Paolo. Paolo asks, how far away are we from developing tailor-made, personalized treatment for people with cancer?
I think we're making great strides in that regard. I think it all began, to be honest, with sequencing of the human genome back in the year 2000. I think discovering all those 20,000 genes has meant that we're able to understand the complexities of cancer. We're able to look at those genes and find new treatments that target the genes in particular. So there are many cancers now, both adult and childhood, where these more personalized treatments are being introduced up front, in the same way as I've talked about today about ALK therapies for neuroblastoma. This is happening more and more, and we're not needing to give as much of the treatment, which is a very, you know, chemotherapy attacks all dividing cells, not just cancer cells, whereas these targeted treatments are directed towards the cells that have the genetic defects which cause cancer. And more these are coming in, and they've all got very long names that we're all having to learn, in the same way as we're having to learn about different antibody treatments. But it really means that personalized medicine is becoming a reality each year for a lot of cancers, including neuroblastoma, which is very, very pleasing to report.
Professor Deb Tweddle, thank you for giving us tonight's fascinating Public Insights lecture. So thank you. If it was live, you'd be hearing applause and we'd be taking you out for dinner. So you'll just have to imagine it. And thank you to our audience for coming along. We hope to see you again. Our next Public Insights lecture is Thursday, the 24th of February. And it's our LGBT plus History Month lecture, and it's given by E.J. Scott. So if you're free, we hope we will see you there next time, and enjoy the rest of your evening.